our species, but other unicellular species. There is a type of vacuole called the contractile vacuole. We'll talk about osmosis at the end of this chapter, but unicellular organisms have a really hard time maintaining an osmotic balance. Their cells want to take in water, and if your cell takes in too much water, it can become so engorged that it actually breaks apart. And so one of the things that contractile vacuoles do is work as like a sump pump, where they're bailing water outside of the cell. But they can also be used for movement. If you contract that vacuole fast enough, you can actually cause the cell to jet forward in space. Okay. Questions about vacuoles? Then we're going to start talking about wholesale cell movement. And the first of which is amoeboid movement. So inside our plasma membrane, and I'm just going to draw an amoeba on the board. Amoebas have a fluid shape. They're really just an individualized cell, right? That has a plasma membrane, no cell wall, so its plasma membrane can go any place that it wants. Inside the entirety of the plasma membrane, there is a network of actin filaments. I'm just going to draw that as a series of blue lines inside here. Like that. And if we were to take this and we were to magnify it, what we would see is plasma membrane and a crisscrossed network of actin filaments. And then incorporated into those actin filaments, there are myosin motor proteins, which I'll draw in yellow. Like that. So if an amoeba, and here's an amoeba right here, and what's happened is, right, it's found some organic material right here that it really wants to consume. And so it's extended out these little projections called pseudopods. And the way that it does that is it causes these actin filaments to push against each other so that you get a directed movement outward. And all of those actin filaments begin moving out in that direction. So you increase the space between adjacent actin filaments. And that's going to cause a pseudopod to form. And then what happens, if the cell wants to entirely move in that direction, what happens is the microfilaments on this side of the cell all contract, and you decrease the space between actin filaments. And it causes a pressure over here that pushes all the cytosol in that direction. So inside our cells, or excuse me, inside our bodies, we have amoeboid type cells that move about, and those are the white blood cells. And you really can't think of them like amoebas that are moving through our bloodstream trying to find invaders, or through our lymphatic system that are trying to find bacterial invaders. And once they do, they consume those bacterial invaders with pseudopod-like structures. Now, another way that microfilaments are used inside our body is with muscular movement. So if you look at cross-section of our muscles, it looks something like this, where there is an anchor protein that extends across the entirety of a muscle cell. And then associated with that anchor protein, there are actin filaments that occur in parallel bands like this. And you're going to get these actin filaments that extend from either side. Cool? But they don't occur in isolation. You actually have multiple groups of these anchoring proteins and myosin proteins. So right adjacent to it, you're going to have another complex that looks like this. But there is intentionally space in between adjacent complexes. And this is how your muscle basically looks when it's relaxed. And then incorporated in between all of those actin filaments, so these are actin filaments, there are myosin motor proteins that extend past that that space in between. And they look something like this, where they have these little fingers that come up and attach to the actin filaments on both sides. That's myosin. So when we receive some stimulus that our muscles need to move, what happens is these myosin motor proteins begin to walk along the actin filaments, and they're actually going to pull these adjacent bands of actin filaments close together, and that's a contraction. So when I move my fist up to my face like this, what's happening is all of the muscle fibers inside my bicep are contracting, right? And all the spaces in between adjacent muscle fibers are becoming closer, right? And the way that our muscles work is those myosin motor proteins aren't going to push my fist back away. Rather, we have complementary muscles that work antagonistically with each other. So I have a bicep that pulls my fist up toward my face, and we have a tricep that's going to cause that to then fall back, right? So in zero gravity, if I was to do this, then my fist would just remain up here until I contracted my tricep, which pulled it back apart, okay? So actin filaments and myosin motor proteins also accomplish muscle contractions.
So if you were to tear your muscle, is it the myosin that's ripping, or is it all of the myosin and actin filaments?
That's a wonderful question. So let's talk about this in the context of strength training.
Mm-hmm.
Because strength training, what we're doing is we're intentionally causing muscle damage in specific regions of our body, right? So that we can increase the mass of muscle and potentially increase strength.
Mm-hmm.
And so generally what happens when you're causing damage is you're causing damage between cells, not within a cell. And you have little rivets, basically, that hold adjacent cells together. And generally with muscle tears, that's what's happening. You're tearing cells apart. And so when you tear cells apart, you're going to be inserting new cellular material in between those adjacent cells. What's really interesting about strength training is strength is a product of the surface area of your muscle. So if I was to take a slice of my bicep, right? The strength of my bicep is really a measure of the cross-section of that area, or a two-dimensional surface. The problem that all bodybuilders contend with, though, is body mass increases as a product of volume, not surface area. And you reach a point at which there is some diminishing returns, where the amount of volume that you have isn't increasing at the same rate as the strength, and you're gaining way more mass than you are strength. Yeah. Okay, does this make sense? So I'm glad that it does, because there's a really good chance that that's going to end up on the exam. Cool. Like, I might have you replicate that diagram. Yes, Matt?
So what does each of these diagrams mean? What is that?
So if I was to, so this is amoeboid movement.
Okay.
Yeah, and that was muscle contraction.
Okay, that's what I was thinking.
Yeah. Okay, questions about that? Okay, very good. So that's not the only way that cells are able to move in space. We also have cellular swimming, and that's accomplished either by flagella or cilia. As I mentioned, flagella and cilia arise from what's called the basal body, which is a manipulation of our centriole. If you take a cross-section of the basal part of the basal body that comes off of the centriole, it actually has what's called a 9+2 arrangement, where you get a centriole, and I'm going to draw a very simple centriole like this. And then coming off of the centriole, you have what's called the basal body that has a 9+2 arrangement. And if you were to take a cross-section of the basal body, it looks something like this, where there are, again, 9 microtubules, but instead of occurring as triplets, they occur as couplets. So there are going to be 2 microtubules attached to each other, like this. But then that +2 refers to 2 microtubules that extend up the center of the base of the body, and really out into the flagellum and the cilia. Okay. So what happens is in between these couplets of microtubules, we have motor proteins that are attached like this. And those motor proteins are also attached inside to these anchoring microtubules. Like that. Now that 9+2 arrangement extends up throughout the length of both the flagellum and the cilia. So the question is, how do flagella and cilia accomplish movement? So if we were to take this structure and we were to rotate it vertical, so this is kind of coming out at us from the board. You had a long flagellum like this. I should drop the curtain. We have some long flagellum. What happens, right, is we have those microtubules that are extending down the middle. That's one of these two right here, and then microtubules that are extending along the sides. Like that. And then we have motor proteins that are attached to adjacent couplets. So when we move a flagellum, what we're really doing is we're moving the microtubules on the left side relative to the microtubules on the right side. We're going to push one up and pull one down. So if we push this side up, and we pull this side down, what's going to happen to that flagellum? It's going to flop over which way, to the right or to the left? To the right, yeah, it's going to go over this direction. Conversely, if we pull the left side down and push the right side up, it's going to cause it to go the opposite direction. So a flagellum is just a long tail that sticks out into some aqueous medium, and it just flops back and forth like this, kind of like a snake's tail, right? It just kind of moves back and forth. And it facilitates movement, but the directionality and the intentionality of that is not very good. Cilia, there's generally a single flagellum on a cell, but there are generally lots and lots of cilia. So with cilia, you get tons of these types of structures that are distributed across the surface of the cell. And when they all beat in tandem, the directionality of that movement is greatly improved. So we talked about flagella with prokaryotic cells as well. And you'll remember the structure of the prokaryotic flagellum is quite different than this. With the prokaryote, there was a motor protein attached to the end of the tail, and it just caused that flagellum to spin in space. This one is a cytoskeletal projection. So this is actually contiguous with the inside of the cell. You have cytosol that's extending all the way up into the flagellum. The evolutionary paths that both the prokaryotes and eukaryotes took to arrive at flagella is quite different. Okay. So this shows you that basal body that extends up into this flagellum, right? And you can see the cross-section of the flagellum has those couplets of microtubules that are all attached with motor proteins. So sperm cells are a great example of cells with flagella, right? When they are swimming to the egg cell, they're just using that flagella to beat back and forth as quickly as they can. Cilia are quite different. They're generally distributed across the surface of an organism. Okay, so please keep in mind, when we're accomplishing amoeboid movement, we're doing that with actin filaments. And when we're accomplishing muscle contractions, we're doing that with actin filaments or microfilaments. The movement of cilia and flagella is accomplished by microtubules, right? Microtubules also accomplish intracellular movement. Microtubules really represent paths or highways within the cell along which we can move and distribute substances. So I mentioned before that there are two motor proteins associated with the microtubules. One is a dynein, and the other is a kinesin. So dyneins transport stuff towards the origin of the microtubule, and this is the origin of the microtubules, right? So if something is being transported towards the centriole, then we're going to use a dynein to accomplish that. If we're moving it away from the centriole, then a kinesin is going to accomplish that. This is what intracellular movement actually looks like. So there's a microtubule path that represents really a highway within the cell. And then there is this motor protein associated with the microtubule. And then there is something called a connector complex, which is a series of proteins that really latch onto whatever it is that we want to move around. And in this case, it's a vesicle.